So if you want to be at the Lunch and Learn for the Parkinson Network of Mount Diablo, the talk today is with Peter Ralston of UCSF Medical.
Thanks, Rick. I'm Peter Ralston, as he mentioned, and it's nice to present to this group. We've been working through all the COVID delays to get this, and the day has actually happened. So as Rick mentioned, I'm an emeritus faculty member from UCSF. I ran a research lab there for many years, and I also directed the medical school course in neuroscience for medical students, and we also taught, of course, graduate students as well. And in retirement, I've had the good fortune to be able to continue to teach for Osher Lifelong Learning Institute, OLLI, which I'm sure many of you have taken OLLI classes at Berkeley, Santa Clara, and Cal State University, San Francisco. And today I'm talking about Parkinson's disease, and we'll get started there. So this lecture will have a couple of dozen slides, and in the slides, I'm going to violate the fundamental rule of showing slides, which is not to have a lot of data on the slides. I can do this because I'm going to give you all the slides after the lecture as a PDF, not PowerPoint. So you can peruse these. And so sit down in the evening with a nice libation, whatever you enjoy drinking in the evening, and you can go back through the slides. So the other thing is on your lecture notes, you have my email address. Rick distributed those lecture notes a few days ago. And if you want to write directly to me at my email address, you see my email says Henry Ralston, not Peter Ralston. That's an idiosyncrasy of my family. None of us are called by our real names. My real name is Henry, but I'm called Peter. I'm sorry, that's the way it is. So that, but don't be confused. Peter Ralston and Henry Ralston are the same person. And as Rick mentioned, we'll take any questions that you put in the chat at roughly 12:45. So some of you have heard some of the information I'll be talking about, because I know there's been a series of lectures on various topics. And I'll try to, my job is to present this material in a way which is clear. And if it's not clear, please ask me about it at the end. So you mostly know James Parkinson, an English physician, described the shaking palsy, as he called it, more than 2 centuries ago, actually. And Parkinson's disease is more than a movement disorder. And this picture actually is in your lecture notes. Many years before the onset of the motor symptoms, the movement symptoms, which are diagnostic for the disease, many people have problems with GI or smelling or various other symptoms that later on turn out to be related to their Parkinson's, which can develop decades after these earlier symptoms. So that's an important thing. It's a multi-system disorder, finally manifested in some sort of movement disorder. It's very common. This is the second most common neurodegenerative. That's a disease in which nerve cells die. Neurodegenerative disorder in the United States after Alzheimer's disease. And this will note says roughly 50,000 new cases a year. The typical incidence is in individuals older than 60, but there are early onset. I've never seen anybody as young as 2, but I have seen people in their 20s. And often that diagnosis is missed because it's so uncommon to have somebody in their 20s manifest symptoms we usually associated with older people. So it's a very common disorder. The disorder is based in the motor portions of the disorder, the movement is associated with loss of the neurotransmitter dopamine, which dopamine, and I'll show you more about this in a second, is a chemical transmitter between nerve cells. And the nerve cells in Parkinson's disease that release dopamine degenerate and die. And the cause of this is not known in the vast majority of cases. The vast majority of cases occur randomly in the population. There are relatively uncommon forms of inherited Parkinson's disease, where a genetic predisposition to the disease has been identified. But the vast majority of people who develop it do not have an obvious cause for the disorder. The basal ganglia, the basal ganglia are structures deep in the brain. This is a semi-transparent view of the brain. So the brain sits in the skull, obviously. The individual's nose will be to your left, back of the head, the top of the head. And deep in the brain, there's this huge aggregate of nerve cells. And these nerve cells are called the basal ganglia. It's an old term, or it goes back actually several hundred years. So we're stuck with it because it really doesn't really help in understanding. But the basal ganglia are associated with a great many functions, including movement functions. And it is those movement functions that are affected in Parkinson's disease. If we now cut away the brain and the brain has two hemispheres, half brains. We cut half brain, remove, in this case, the left half brain, and look at the medial surface of the right half brain. Again, the nose is here. Deep in that brain is a structure called the brain stem, which connects to the spinal cord. And in that brain stem is a cluster of tens of thousands of nerve cells, which produce dopamine. And they're in a structure called substantia nigra. You'll see why it's called nigra, because it looks black in a fresh brain, and a couple of other regions as well. Those nerve cells have axons, which we'll talk about in a second, long processes which convey the dopamine throughout the brain, including into the basal ganglia. Sorry, my advances. When you cut the brain stem, this would be two autopsy specimens. Let's just go. If we cut through the brain stem right here, we're cutting through a cylinder, which is the brain stem. And now looking at that cylinder, we see here the cut surface of the brainstem where you have a structure called substantia nigra. And that substantia nigra has axons, processes, which carry dopamine to different structures deep in the brain, including structures in the basal ganglia, okay? It is these cells in particular that degenerate in Parkinson's disease for reasons we usually don't understand. Therefore, the processes called axons of these substantia nigra neurons degenerate because their parent cells degenerate. So you lose the input from this structure to the basal ganglia. These are autopsy specimens. This is a brain from a person without Parkinson's and a person with Parkinson's. Substantia nigra, these are fresh cut brains in autopsies. You can see it looks black. So nigra, black. And that's because of pigment in these cells, in the cells that produce dopamine. So when those cells die, so does the pigment. So you can actually see here the loss of that pigmentation in normally in the cells which normally would be producing dopamine. This degeneration of dopamine-containing cells in Parkinson's can begin 20 or 30 years before the onset of the movement disorder which we associate with Parkinson's. And it is estimated that one has to lose about 80% of his or her dopamine-producing cells before you actually begin to manifest the symptoms of Parkinson's disease. So this can be going on for many years before one ever gets any symptoms. Now, if we look at nerve cells, and this is just a schema of a nerve cell, we have hundreds of billions of nerve cells in our brains. In the substantia nigra, there are several tens of thousands. And these nerves, all of our nerve cells have a cell body, and where we have the DNA, which drives the processes of protein synthesis and everything else that cells do. So processes arising from the cell body called dendrites, dendrite just means branch, where incoming inputs contacts this and activates the cell. And then the cell sends signals out the process called the axon. Remember, I talked to you about the axons carrying dopamine to wherever they were sending into the brain. So the axon is a single process that ends, well, eventually in contact with the nerve cells, okay? So this axon actually carries electrical signals along its length, and the length can vary from a few millimeters to a whole meter in the case of motor neurons of the spinal cord. So a varying length out to the synapses where in this case, dopamine would be released on the next nerve cell. Now, usually when I would give these lectures, I would give it with a whiteboard. And of course, with Parkinson, with I'm sorry, something's going on. Okay. My movement between slides is not what it should be. So the axon comes and forms a ending at a structure called the synapse. So the axon then had an electrical signal entering the terminal. In the terminal are little packages of transmitter, in this case, dopamine. When the electrical signal enters the axon terminal, those little packages of transmitter blown up here in this part of the slide, and each one of these green structures we can think about as dopamine-containing vesicles, then are liberated and travel in less than the thousandth of a second. This happens very rapidly to combine with receptors for the transmitter, dopamine in this case, to activate the next cell. So in the disease Parkinson's, there is less dopamine released because many of those cells have died. And there's less activation of the postsynaptic receptors for dopamine. So treatment would be to give a dopamine precursor, L-DOPA, D-O-P-A, which many of you know a great deal about, which actually is a precursor to form dopamine. And you can also give synthetic molecules which act like dopamine on the receptor. And those drugs in Parkinson's help to activate dopamine receptors in the absence of normally produced dopamine because of the death of dopamine-producing cells. So a frontline treatment for Parkinson's would be to replace the lost dopamine by a metabolic precursor of dopamine, L-DOPA, or synthetic molecules to activate dopamine receptors. Another important aspect of this, which I'll come back to, is here's a nerve cell with its axon going out to the axon terminal. In addition to electrical signals, which go out the axon, there are also movement of molecules down the axon from the cell body to the terminal, and from the terminal back to the cell body. And normally if the cell here is producing dopamine, the enzymes that synthesize dopamine are produced in the cell body, travel out to the terminal, where they then help in the synthesis of the transmitter, which is then released. A very important aspect, we know that materials can travel backwards to the cell body. And we're going to come back to this in a few minutes when we talk about misfolded proteins, alpha-synuclein, which can be synthesized in, say, the intestine, which are taken up by the terminal and come back to the nerve cell and actually kill the cell. So, and there are viruses like rabies virus, poliovirus, they also can travel in backwards up the axon. So there's communication normally between the cell body and the terminal back to the cell body, and a little bit of called molecular motors to move these substances along the axon. So in the neurodegenerative diseases in general, a major advance in recent years has been the recognition that most of these degenerative diseases of the nervous system are caused by misfolded proteins of various types that get into nerve cells and kill the cells. And in the case, and they've been identified in Alzheimer's, and I'll show you a slide of that in a second, and other sort of dementing disorders, and in the one we're talking about today, Parkinson's disease. So those misfolded proteins are thought to be the root cause of many of these disorders. So they tend to accumulate as we get older. And as we all know, age is an increasing risk factor for every damn thing you can think of. Protein misfolding and aggregation then inside cells. Occasionally, there are genetic forms. As I mentioned, a tiny percentage of people with Parkinson's actually have demonstrable genetic disorders leading to that. The vast majority do not. And prion disease, also a misfolded protein, is the only known infectious transmissible form of these diseases. Prion disease, for instance, remember mad cow disease. It is a disease of cattle, which actually is, again, a misfolded protein. If we ingest that, it can sicken us, okay? You may remember 20 years ago in England, there were breakout of mad cow disease. They had to ban all beef production in England for several years to root out the diseased cattle. So people didn't eat that infectious disease. The vast majority, Alzheimer's, Parkinson's, and the like, there's no evidence that they are infectious. So here now are examples of disorders. I'm sorry, I don't know what in my, okay. The alpha-synuclein is related to a group of proteins called synuclein, which can become misfolded. And they are thought to be major participants in Parkinson's disease, okay? Alzheimer's disease is related to a different molecular structure called tau, which also can become misfolded and be related to several other different diseases, as can altered structures of synuclein. Okay. Lewy bodies are aggregates of alpha-synuclein in nerve cells. They are found in two major diseases. There's one, primarily a dementing disorder called dementia with Lewy bodies, and Parkinson's disease. And it turns out these two diseases are related. There are two ends of a clinical pathological specimen. And in dementia with Lewy bodies, there's a significant involvement of the cerebral cortex, as you might expect if one has death of many nerve cells of the cortex where our cognitive functions are located, one can have dementia. In Parkinson's disease, the pathology is most in the basal ganglia, which we've been talking about, not in the cerebral cortex. But if one does a spectrum, it's a bell-shaped curve with some people being at one end only with Lewy bodies in the basal ganglia, those, and therefore only with movement disorders and the other signs of Parkinson's disease. And then some only with Lewy bodies in the cortex and only dementia, and some with a mix of the two. So that's why roughly, and it varies with the study, roughly 20% of people who start out with the motor side of Lewy body disease will go on to have dementing disorders as well. Others with Parkinson's disease will never have any dementia associated with their disorder. So here you have a normal alpha-synuclein. Alpha-synuclein is found in normal brains. It's found in synapses. That's why synuclein. The synapse is that structure where the transmitter is released to activate other nerve cells. And its function is largely unknown in normal brain, although there's some suggestion that it helps in certain forms of transmitter release. So we all have normally folded alpha-synuclein. Then for reasons we don't understand, in most cases, they can begin to misfold and clump together to form larger and larger aggregates. Normally there is the small size, but they begin to clump and aggregate together and eventually form the structure called a Lewy body, which is a pathological finding in in Parkinson's disease. So Lewy bodies are an abnormal aggregate of abnormally folded alpha-synuclein protein molecules. We now also know that Lewy bodies can move between nerve cells. So given population of neurons that have become infected, and I'll talk about ways in how the initial infection might occur in a minute. So they accumulate, here we have these little dots are normal alpha-synuclein. They become aggregated in these red curly structures and can become taken up by other nerve cells, which can seed the production of more and more aggregates, and ultimately then end up in destroying this nerve cell and also influencing a structure called microglia. These are nerve cells which signal information. Microglia are supporting cells in the brain. Glia just means glue. So small cell that has these functions. And microglia then are now known to be important in the generation of the abnormally folded alpha-synuclein. And I'm going to talk about a couple of drugs at the end of the lecture, which are trying to target this abnormal microglial cell, which produces these abnormal aggregates of alpha-synuclein. A discovery which actually occurred first of all in humans, I'll talk about how that happened in a minute, is that abnormal alpha-synuclein can be formed in the intestine. And these obviously are mice and rats where the studies have been done. And there's actually a mouse model of Parkinson's disease that exhibits some of these symptoms of human Parkinson's disease. They have trouble initiating movements and they actually have tremors. And it was found out that if you take these mice that are models for Parkinson's who have movement disorders and give them high doses of antibiotics and kill the gut bacteria, that they have reduced symptoms. Okay. And then if you take fecal transplants from humans who have Parkinson's disease and take those fecal transplants and put it into the guts of these mice, they will have return of their movement abnormalities, as if something is going on in the gut that yields this abnormal disorder we call Parkinson's disease. Now, how did this even get understood? About 30 years ago, a study began in Sweden looking at people who had chronic ulcers, chronic gastric ulcers. Our stomachs produce hydrochloric acid. Hydrochloric acid is produced when synapses from structures in the brain called the vagus nerve come to the stomach, and when they fire, they induce the formation of gastric acid. So beginning about 30 years ago in Sweden, a group of people, and actually it turned out to be several thousand over several decades, had the vagus nerves cut in order to improve their likelihood or reduce their likelihood of getting a I'm sorry, I don't know what my computer is doing, but it's to decrease the synthesis of gastric acid. Then when these people were assayed, and Sweden has a universal health plan, which we don't, they track patients all over the country, which we don't. And they found that incidentally, there's a decreased incidence of Parkinson's disease in those individuals who had their vagus nerves cut many years before. And people began to look at this mouse, which they knew tend to develop Parkinson-like syndromes. And they found when they cut the vagus nerves in a mouse, they reduced the incidence of Parkinson's disease in those Parkinson's symptoms, like in that mouse. So that's how, just by chance, people began to look at the intestine. Coming back to this picture I showed you earlier, if we imagine that this now is a vagus nerve cell, it goes to the stomach and the intestine actually to induce activity in the gut. If the mouse or in the human is producing alpha-synuclein in the gut, it can come backwards up the axon into the nerve cells. And if you cut those axons when you treated these people for their gastric acids, or when you took an experimental mouse, even though they may have alpha-synuclein being produced in the intestine, it can't get back because you've cut the axons to the intestine. So this finding in humans, when they weren't looking for that at all, they're just trying to treat gastric ulcers, resulted in a hypothesis that a major source of alpha-synuclein in the brain was from the gut. Okay. Now let's look at a couple of ways that we can try to treat people with Parkinson's disease. In addition to the drug treatments to replace dopamine or to activate dopamine receptors with synthetic drugs, which work on dopamine receptors, you can also put electrodes into the brain to stimulate areas of the basal ganglia associated with Parkinson's disease. In other words, stimulate nerve cells directly that no longer get the innervation from the dopamine. The dopamine's gone or largely reduced. And you can put electrodes into the brain in a surgical procedure. These are very fine stimulating electrodes which go into the brain, but it requires neurosurgical procedure. You make a small hole in the skull and you put an electrode in the brain. This has been done in a few hundred people. It's called deep brain stimulation. And I think some of you have already had a talk about deep brain stimulation. But I'll show you a couple of pictures, slides of people who've had deep brain stimulation. This is at the San Francisco VA. This individual has a disease that's different than Parkinson's, but has a similar pathology in that they have trouble initiating movements. So this is not a Parkinson's patient, but it is a patient with deep brain stimulation. So first of all, here's the patient before they have deep brain stimulation. And they're asked to gradually get up.
Stand up from the chair if you can.
And you can see they have really hard time moving, okay?
You should take some steps over towards the camera there.
And just these very short steps moving, okay? Now, this is the same person. I'm not cheating. This is the same person following deep brain stimulation. So they greatly improved, obviously, the motor skills of this patient. It's quite nice. The trouble is there are more than 1 million people in this country with Parkinson's disease. And you can't possibly carry out, it's very expensive. This is done at the VA. It doesn't cost the patient anything. And it's way beyond anything we could do to help people with their motor disorder of Parkinson's disease, because it's, you know, it's a big deal to do this, and you can't treat 1 million people. But I just wanted to show you that deep brain stimulation can work for motor systems disorders in people with this type of rigidity. Okay, there's another technique which has some possibility of being more widely distributed than deep brain stimulation, but still is not an answer for the millions of people with Parkinson's disease. And that is to substitute dopamine-producing cells, to generate them outside the person's body, and then inject them into the brain. Okay, this technique is a brain implant, and it's actually been going on since the 1980s. Initially, they were taking aborted fetuses, removing dopamine cells from aborted fetuses and injecting it into the brain. Okay, obviously, this is not going to work in a big scale. The whole issue about the ethical issue, these were spontaneous abortions, but just the whole issue of aborted fetuses, that wasn't going to work. But in the beginning, in the early part of the 2000s, and this led to a couple of Nobel Prizes, was to take cells that, say, from the skin, that are normally are stem cells deep in the skin, and make them, they call into what I call pluripotent stem cells. In other words, they're not just stem cells for the skin, they can be stem cells for anything. And take a person's own skin cells, convert them into induced pluripotent stem cells derived from skin, and have them make dopamine, and then inject them into the brain of that patient. Number one, you don't get the immune response because they're the patient's own cells. They can now make dopamine. And this is a study in the New England Journal of Medicine a couple of years ago, actually it's a year and a half ago. And this is this particular patient. They're just looking at the disease, Parkinson's disease quality of life. Many of you have probably taken this questionnaire. The higher the number, the poorer, the worse the quality of life. And these are months since the implantation. So initially, the patient reported, and I don't know because it's anonymous if it was a male or female patient, if her symptoms got better, and the caregiver could participate in this report as well, then began to backtrack, and then a second implantation was done. And the symptoms got better again based upon this questionnaire, and stayed in this very good report out to 2 years. So this is 2 years after the first implant and 18 months after the second implant, okay? This, again, is a procedure which is not an answer for 1 million people. It certainly helped this individual, and now obviously there's more people being studied for longer periods of time. So far, the reports are out for 2 years. Of course, this person's going to be assessed farther out. So the idea is you substitute cells which have now been induced to be able to make dopamine from these individuals' own cells, put them into the brain, and they greatly ameliorate the symptoms of the disorder, okay? Oh, God. Okay, there's another drug. I apologize, my PowerPoint usually doesn't do this to me, and I apologize for it. That are targeted now, and they are in clinical trials. There are 2 different drugs I'll talk about. And at the bottom, you can see UCSF is doing some of these clinical trials. And many other institutions are as well. And you'll have access to this URL if you're at all interested in clinical trial presentation. Okay, here's a drug called NLY01, which acts on these glial cells, the microglia. The microglia turn out to change the properties of other glial cells, which then produce some sort of a toxin, which is unknown, to lead to the accumulation of alpha-synuclein in nerve cells, killing them. So the drug under clinical trial now acts on these glial cells to reduce their ability to set out this cascade of accumulation of alpha-synuclein in nerve cells, okay? And this is actually a study in Nature Medicine from 3 years ago, and this drug now is under clinical trial at UCSF. I think it's stage 3. I'm not sure about that. I think it's a stage 3 clinical trial. Another drug, which basically the same idea, and these are in the mice that are a mouse model for Parkinson's disease. And when they have injected alpha-synuclein monomers into the brain, if they have not received this drug, which is spelled out down here, they go on to produce all the alpha-synuclein aggregates, which accumulate in nerve cells and kill them. But the drug, when the drug is administered to these mice, and then you inject the alpha-synuclein monomers, they do not get into this cascade of producing huge aggregates of misfolded protein. So this drug does not act on the microglia, it acts on the molecular mechanism which produces aggregates of alpha-synuclein. The other drug I talked about acted on the microglia, but this again, both of these drugs are trying to act on the fundamental cause of the disorder. That is the aggregation of alpha-synuclein in nerve cells in people with Parkinson's disease. Okay. So I apologize for the problems with the PowerPoint. I'll just have to find out what's going on with that. Dick, can we come back to what about? Oh, I'll stop screen share. There I am. Okay. Now there's 2 questions so far in chat. And I just have to get my glasses. I'm sorry to read them, and you're welcome to put more questions in chat.
Okay, so when people want to ask questions, they will have to unmute themselves.
Yeah, thank you. Yes. Okay, this is Janice and Derek Ransley. Is there any normal alpha-synuclein in the CNS at all? Okay, that's a very good question. Yes, there is. Alpha-synuclein is the monomer, the non-highly folded protein, is present in our synapses, in the axons of nerve cells. And it plays a role in the release of transmitters normally. And for reasons we don't understand, in those people that go on to get a synucleinopathy. These are proteinopathies. Pathy just means illness. So synucleinopathy is an illness related to synuclein. Those people that get this misfolded aggregates of alpha-synuclein that kills off nerve cells, that starts with the normally present synuclein in axon terminals. Now, what cell produces it in the gut and elsewhere? That's a very good question. To my knowledge, those are intestinal lining cells, some of which are nerve cells. But I don't know if there's a particular cell type which is uniquely associated with the production of alpha-synuclein aggregates in the gut. And some of the earlier work in rats, investigators took laboratory-produced aggregates of alpha-synuclein, in other words, abnormal aggregates, injected it into the intestine of rats and found, and they labeled it with a way you can label the protein, and found that same protein after a few days was now in the brain, having gone from gut to brain via those neurofibers that I talked about earlier. I'm sorry, let's see.
I have a question.
Sure.
Can I ask a question?
Yes, you can. Go ahead.
If a patient has stem cells collected prior to diagnosis or about same time of Parkinson disease, can they help the patient with either motor or non-motor symptoms? Can they be used? Because my husband had them collected because he also has multiple myeloma, so they had collected stem cells. Can they be used? Are they useful or no?
The use of stem cells by major universities is very carefully regulated, and I don't. I guess the best person, I can't answer. I don't know the answer to the question. I guess the best people to talk to would be, say, a neurology group at a major medical center, and say, would donor stem cells be of value to you or not? As I mentioned in the study that I referenced, the stem cells came from the patient himself. Because if you take my stem cells, modify them, inject them into my brain, my immune system does not recognize them as foreign cells because they're my cells, okay? If you take stem cells from Mr. Jones, modify them, inject them, all the immune reactions to a foreign cell would come to play because they're not my cells. So that's one caveat there.
But in this case, these are patients' own cells, though.
Yeah,
the patient was first had multiple myeloma, and the stem cells were extracted, and then diagnosis of Parkinson came. So would they be? Is there any chance it could help?
Yes, there is. If the study that I showed you with pluripotent stem cells was done at Brigham and Women's Hospital, one of the Harvard Medical School hospitals in Boston. I don't know if there's any local group doing induced stem cell work in Parkinson's disease at either Cal or UCSF or Stanford. I just don't know. But the best way to find out is to ask, say, a neurologist from one of those two institutes or Davis, if that work is going on. It is definitely going on in Boston. And it is going on at Hopkins and a few other places as well. Induce stem cells to make them produce dopamine. But the answer is yes, it is possible. But I don't know if it can be done locally or not. I just don't know.
Thank you so much. Thanks.
You're welcome.
Question. Do the induced stem cells, will they then clog up with the alpha-synuclein later?
That's a very good question. And they haven't been gone long enough because the whole disorder, as we saw from the progression from non-motor symptoms to motor symptoms, can occur over decades. And the work with induced stem cells is only 4 years old, okay? And so whether or not, and those cells were still producing dopamine, which means they're still alive 4 years after they were put in the brain. Whether or not some of them have now been altered by alpha-synuclein, we just don't know. So the answer is we don't know. It hasn't been going on long enough to find out.
Thank you.
Yes.
All right, we have a question from Dennis Krause. Do you feel that the use of over-the-counter probiotic capsules is likely to be beneficial? And that obviously relates to the gut connection with Parkinson.
Yeah, that is very interesting. I have no idea. There's been quite, this whole thing of probiotics is again very big. It's a way to sell something, and we want a diverse microbiome in our cuts. There's no doubt about that. There's a real problem if you take high dose antibiotics for some infection, you wipe out a lot of the normal microbiome of your gut. You can get into real trouble from that, and restore it by taking some sort of yogurt or something like that. But there's, to my knowledge, there's no good study that shows that a particular line of intestinal bacteria is more beneficial than another line. We tend to adopt over our lifetimes a line which is beneficial to us and may or may not be to somebody else. It doesn't hurt to take a probiotic which doesn't have infectious bacteria in it. But whether or not it would be beneficial, I don't know. It doesn't hurt to try that. Yes, sir. James Blackwell, you need to unmute yourself. You know, on the bottom left corner of your screen, you click on mute.
Can you see that, James?
Is that working?
Yeah,
There you go.
that is. You're live now.
Thank you.
A little louder.
I'm Jim Blackwell. I'm a FACP internist, and I've had Parkinson's about 10 or 12 years. Let me first say this is the best lecture on Parkinson's I have ever heard, period, bar none. I just wondered if there's any way to listen to you further or get a podcast or something so that I could listen to your lecture again and again.
Well, Jim, that's very kind of you to say that, especially after I screwed up on some of the slides. The lecture that I just gave is recorded, and it will be released, and you can find out about that from Dick.
Yeah, it will be released within 48 hours.
Good. Okay.
And it'll be on the PNMD website.
Yes, it'll be on the PNMD website, and our IT person will also send a constant contact email to those on our mailing list with the link. And the link will also let you look at the slides and the lecture notes as well.
Yeah, these slides will be available to you starting probably tomorrow. You can look at those.
That's great. Thank you. We're new to this group. So that's wonderful to know that it's recorded and there are all those other resources there. Okay.
Thank you, Jim.
Thank you.
Are there any other questions?
Peter, I have a question.
Yes.
Is neuromelanin structurally or metabolically different than peripheral melanin in the skin?
Good question. Neuromelanin is biologically very similar to, but not identical with cutaneous melanin. Melanin is a skin pigment that we all have in greater or lesser degrees. And it is produced by particular cells in our skin and many tissues, obviously. And neuromelanin is biologically, molecularly very similar to, but not identical to cutaneous melanin. And it is associated, and I never have really seen a clear explanation why dopamine synthesizing cells of the basal ganglion, of the substantia nigra also contain neuromelanin. I'm sure there's a biochemical reason for that. I just don't know what it is. But
I'm curious about that because there must be some interaction between the dopamine production and the pigment because it correlates with symptomatology. And
what I should do is go back and see if I can find out a good source to answer that question. I haven't searched for it adequately, obviously, otherwise I'd be able to answer it better than I have. But it is present in the dopamine synthesizing cells. And a very close relative, cutaneous melanin, is present in our cells of our skin and many other cells that produce pigments.
I've read a number of articles about genetic engineering and the relationship to LRK genes, LRKK genes, and I have to claim that I really don't understand how genetics can help us improve the life of patients with Parkinson's. Could you possibly explain that?
If, when a person presents, as I mentioned, with all of a sudden you realize that either their physician or the individual, that I would likely have Parkinson's because of my movement or whatever. The vast majority, at least 80% of the dopamine synthesizing cells of the substantia nigra have disappeared. And now with genetics, if you can, it's really a way to alter our genes when you do induced pluripotent stem cells. You're changing those cells of my skin, which normally would not produce dopamine, to cells which then can induce dopamine. So that's a genetic change in those cells, and then put them into the brain. So that's one way to help, because you are increasing the number of normally functioning dopamine cells. In other treatments, for instance, if you take L-Dopa or other drugs for your Parkinson's, it helps for a while, but as you all know all too well, many times there's side effects, and it does not, those drugs don't cure the disease. They help the symptoms for a while, greater or lesser number of years, but they don't cure the disease. If you could actually change the inject enough induced cells and to help people over 20 years, they actually are replacing the lost cells. But you may not be getting at the real problem if it's going on in the cerebral cortex, for instance. There may be cells that are producing aggregates of alpha-synuclein, which are killing cerebral cortical neurons. And that, because you gave dopamine cells in the basal ganglia, that's not going to change what's going on in your cortex. So if this was a disorder with a single gene deficit, like Huntington's disease is a terrible neurodegenerative disease, it's rare, but it's due to one gene being altered. And you can actually replace that gene now with a new CRISPR technique, gene editing technique that Jennifer Dowda got the Nobel Prize for. There's work that may be a way for genetics to cure a fatal disease. Because you have one gene, if you can repair it, the disease stops. There's not, unfortunately, if anything, there are multiple genes involved in Parkinson's, and the chance to cure Parkinson's with genetic techniques, at least now, we don't know enough to be able to do that. Whether or not the same, I mean, these things are moving incredibly rapidly, and maybe a decade from now we would, but as of today, I don't think so, because it's not a single gene disorder.
Okay, thank you. Are there any other questions?
Yeah, I have one more question. I wanted to ask, like, we're talking about dopamine. So is there a way, scientific way to figure out how much dopamine, like carbidopa, levodopa, does a patient require at a particular time? Because too much is problem, too little is problem. So is there a way that to figure out what is the optimum dose?
You go to a physician who is experienced in treating people with Parkinson's disease, and it gets titrated for that person. There's no one magic number suits all at all. And it depends where you are in your disorder, how many dopamine-containing cells you've lost, and you work with the physician to get the best result you can. And you're quite right, there are side effects which may limit the ability to titrate this for more or less treatment. But you want to go to somebody who sees a lot of people with Parkinson's disease and is used to working with a very variable patient population, because it is variable. And you can't, the answer is there's not a single dose that is right for everybody. It's just not.
Thanks.
Another question. I was wondering on some of these things that you mentioned, cutting the vagus nerve and putting a stool implant into the gut, and the drugs that act on the glial cells, the drug that acts on the PARP inhibitor. How close are all those things to being something that a patient could receive treatment with?
Well, if those PARP, the last two slides that I showed, those are in clinical trials now. There's an initial report on the ILY drug that acts on microglial cells with a small patient population showing an improvement compared to placebo. That was a stage, I'm not sure if it was a phase 2 or phase 3 trial. So anyway, it depends obviously on the results of good or bad of the present trials. Cutting vagus nerves is not going to be something any of us wants to go through, because by then the alpha-synuclein is already in our brains. So that's not going to be a treatment, because you're shutting the barn door after the horse has gotten in, the alpha-synuclein has gotten into our brains. So the best hope for an effective treatment that would at least improve the clinical outlook of people would be those drugs that stop the production of alpha-synuclein. But, and again, hopefully those cells, it would be given in an early phase of the disorder, when we haven't lost too many of those cells, or it hasn't invaded our brains to give us a dementing disorder, Lewy body dementia, which doesn't help a lot. I think it will help our children. I just don't know how many it's going to help us in our 60s, 70s, and 80s.
Thank you very much.
Dr. Ralston, I have one question. I remember as a young physician, I used to assist in surgery. Of course, the vagotomy and gastrectomy was popular for peptic ulcer disease. I just wondered if anyone has ever looked at that patient population of those who had that procedure done with the vagotomy to see the incidence of Parkinson.
Well, that's the study from Sweden that we talked about, the vagotomy patients. In their group, I mean, I don't know if people have looked at vagotomy patients in the United States. Of course, there's a lot of them, just like in Sweden, but I've never seen a publication from a US group. And as I mentioned, the problem is each institution tends to have its own group in the United States of patients. We don't have a national health service, which pulls all that data centrally. We are behind most of the other Western countries in our healthcare system, as we all know. There's not a uniform system which is available to all patients. And God knows how long that's going to take before it ever happens. Okay. So I don't know of an American vagotomy study that's been published.
Thank you.
Okay, well, we're supposed to, I'm sorry.
Yeah.
Go ahead.
I was just going to say that we're at 1 p.m. Acting as the formal timekeeper here. And I want to say that, first of all, I want to thank you very, very much, Peter. Fascinating presentation. And I wanted to let everyone know again that we will post this within 48 hours on our YouTube channel. We will send out links that'll be posted on our website. But you'll also, if you're on our mailing list, then you will get a constant contact email reminder. If you subscribe to our YouTube channel, then as soon as the video is uploaded, you'll get a notice. And we make every effort to make sure that the critical links, let's say the clinical trials at UCSF, are posted in the YouTube description. So you can simply click on them. Today's Lunch and Learn lecture has been a presentation of the Parkinson Network of Mount Diablo. Please visit us at our website for more information.